Hello and welcome to week 8. This week we have finally gotten to photosynthesis. So we've already talked a lot about photosynthesis in terms of knowing where it's happening in plants or photosynthetic bacteria, and thinking about it as sort of the opposite of what's happening in aerobic respiration, where we take glucose and oxygen, break it down to get out energy, to get out ATP, and as a result, produce water and carbon dioxide. So now we are going to be focusing on the flip side of this cycle of how plants are able to take light energy and convert those carbon dioxide and water molecules into that delicious glucose. So photosynthesis is convenient because the word itself tells us what's going on. Photosynthesis, light making. Photosynthesis is using light to make things. I'll comment photosynthesis, we think about it as all those green leafy trees and grass, but it's important to actually comment on the fact that most photosynthesis is actually happening in our oceans. And we can think about the microorganisms that is actually doing most of this photosynthesis on planet Earth. We call organisms that do photosynthesis photoautotrophs. Photo meaning light, auto meaning self, and troph meaning feeder. So photoautotrophs feed themselves by using light. They feed themselves, as we're going to see, by making sugar from light energy. And I had this in the previous slide, but can you imagine how wonderful it would be if we didn't need to eat and we could just go sunbathe? But alas, we don't. But we get to eat the delicious plants that do. So now onto this picture of thinking about the carbon cycle and what is carbon fixation. So we can think about how we can break those carbon-carbon bonds when we're releasing carbon dioxide into the atmosphere. That carbon dioxide gets converted into sugars by plants in photosynthesis. Then we can just think about what happens to those plants. Well, some of those plants get eaten, some of those plants die, and that carbon dioxide that had been converted into the sugar or been fixed from atmospheric carbon into that organic molecule, well, those organic molecules then get stored. When they get stored for a really long time, that's how they become fossil fuels. That's how it becomes those hydrocarbons, those carbons and hydrogens. Eventually, that hydrocarbon can get pulled up and it can get burned and it can cycle all of these things again. Another way to think about also that cycling is the aerobic respiration done by the plants and animals. I can never decide which graphic to use, so here's another graphic, same idea, but really again pointing out where do those fossil fuels come from? It's organic material. And how did that organic material come to be? How did that organic material get the carbon to make up those organic molecules? From the plants. Ultimately, we can thank the sun and we can thank the plants for bringing in the vast majority of energy into the ecosystem. And we can remind ourselves that, of course, we're never creating or destroying energy, it's just cycling through. Okay, so now we get into the nitty-gritty of photosynthesis. And we can talk about overall what's happening. Overall, photosynthesis is converting the energy of light into the energy of chemical bonds by taking carbon dioxide and water and rearranging those molecules to make up glucose and then release oxygen. We can see that photosynthesis can get broken down into 2 stages. And conveniently, the stages are the same as the name. So the first stage is the photo stage, is the light-dependent reaction. The second stage is the actual synthesis stage, and that's called the Calvin cycle. So you are going to need to know overall the reaction to describe photosynthesis, and then in depth the mechanisms going on in each of these compartments. So just like for aerobic respiration, it's important to know what structures we're talking about when we're talking about photosynthesis. Chloroplasts are the organelle in which photosynthesis takes place. We saw this earlier in the class, but we can think about the structure of chloroplast, and we're reminded of those bacteria we studied, those prokaryotic organisms that actually had almost the same structure because that's where they evolved from. So we have a leaf in this example, and we can see these different cells. We can see particular cells within the mesophyll that are filled up of all these green dots. What are each of those green things? Those are the chloroplasts. That is the organelle with the two membranes in which photosynthesis is taking place. So of course, you're going to need to know a little bit more about the structures. Overall, the whole thing's the chloroplast with those two membranes. And then you've got even more membranes. So for the chloroplast, we're going to see it's these thylakoid membranes that all make up a granum, that there's this stroma that's the liquid in between. And we're going to see that some stuff is happening in these thylakoid membranes and in this thylakoid space. So again, you want to be able to recognize these structures and identify them, and particularly identifying and making reference now to this thylakoid compartment in this space in between these membranes. So again, overall, and I'm saying this many times because you absolutely base level must understand. Photosynthesis takes 6 molecules of carbon dioxide, 12 molecules of water, light energy, and makes glucose, C6H12O6, 6 molecules of oxygen, and then 6 molecules of water. You can see that water is both going to be used as input and is actually produced. And if we actually want to follow the different molecules, we can see how we have those 6 molecules of carbon dioxide and 12 water molecules. Ultimately, we can follow how we get those ultimate products and why we had water as both a reactant and a product. So I said that photosynthesis takes place in 2 stages. There's the photo stage and there's the synthesis stage, photosynthesis. So you are going to be expected to understand things at the base level of this picture, of understanding what is coming into each reaction and what is coming out. Now you're going to need to know more than this, where again, first reactions, the light-dependent reactions, we're going to see that are going to involve, of course, light. And this is where we're going to be splitting that water and producing oxygen as a byproduct. We're going to be getting out these energy currency and these high energy electrons. They're going to be utilized in that secondary synthesis stage. And it's the secondary synthesis stage in which that sugar is actually produced from taking the carbons from carbon dioxide. So before we can explain how the process works, we need to talk a little bit more about energy and light. So energy radiating from the sun travels in what we call photons. So we can think about all the energy coming from the sun as the energy spectrum, and we can think about the light that we're familiar with. That's the visible light. That's the wavelengths that our eyeballs can actually see, that our brain can see. But it's actually a tiny, tiny section of the amount of energy coming from the sun. So there's high energy. These have short wavelengths, and these are things that you've heard of that you can be concerned about, like X-rays. Whereas on the opposite end of the spectrum, we have the lowest energy wavelengths. These are things like radio waves. So people have pretty much heard to be concerned about getting X-rays unnecessarily or getting too much ultraviolet radiation. Whereas people are genuinely not super concerned about sitting in front of their microwaves or their radios. This is because, again, these guys have lower energy. You can see that difference in energy as a reflection of the wavelengths of those different colors. So a wavelength is the distance between 2 peaks, and we perceive each different wavelength as a different color. The shorter the wavelength, so the closer they are together, the greater the energy. Also conveniently, I want to point out that to the left, or on the higher energy side of violet, is the ultraviolet radiation. On the other side of the red is the near infrared and then infrared radiation. So if you want to sort of orient yourself, the nice thing about the visible light is you can put it within the energy spectrum pretty well, just based on the terms itself. So thinking about those terms and thinking about those colors, we get to talk about a pigment. So a pigment is an organic molecule that's going to absorb light at a specific wavelength. It is going to reflect back a particular wavelength of lights, and those are the ones that are going to hit our eyeballs, and those are the colors that we actually see. In plants, we mostly see green, and that's because they're full of chlorophyll A. Chlorophyll A and chlorophyll B are remarkable molecules as far as evolutionarily and what the role they play in organisms. It's interesting to make parallels between hemoglobin, which carries oxygen in animals, and chlorophyll, which we're going to see in a moment, is going to have this special role with electrons in plants. I'm not going to play this right now because you're watching me do a video, but I would say I would highly recommend you click on this video in terms of describing why plants are green. I would also recommend you watch this video, but we can think about the fact that plants are full of all these different pigments, and each pigment is going to absorb different wavelengths of light and reflect back. So we can actually look at the absorption spectrum of different types of pigments and see that that's how we see the different colors of leaves. When the chlorophyll A's and the chlorophyll B's go away in the fall and we're left with just the carotenoids, then we see all those beautiful oranges and reds and yellows. Okay, so as our last preview slide, again, we're going to be talking now in depth about the two different reactions. The light-dependent reactions, which we are going to see are going to take that light energy and produce high energy products, ATP and NADPH. And then we're going to have this secondary stage that's going to use those products, use that energy to do the building of the sugar. At the primary level, you need to understand what's coming in and what's coming out. So you need to know the reactants and products in the light-dependent reaction. So we have the sun. The sun is radiating all that light energy. Some of that energy is going to be used within the chlorophyll molecule, within the chloroplasts, to excite an electron. But other of that energy is going to be emitted off, is going to bounce back off and be re-emitted at a longer wavelength. There's less energy, and some of that energy is also going to dissipate as heat. But we're going to focus in on what's happening in that chlorophyll. So I've got a whole bunch of different pictures here. The key is for you to figure out which makes sense and to use that as the model in your brain. So again, we're talking about chloroplasts. We are going to be zooming in onto, again, the thylakoid membrane. And what is happening within that thylakoid membrane? Within all those phospholipids, there are these chlorophyll molecules and photosystems. These chlorophyll photosystem protein molecules are embedded physically into the thylakoid membrane. So I'm going to make a comment to remind ourselves a little bit about electrons. Now we remember that there's those different orbitals, there's those different shells where the electrons can go, and you can move an electron into a higher shell, into a higher state that requires more energy. And then as that electron returns back to its normal state, it's going to dissipate that energy and it's going to go out as heat or release that color, which is called fluorescence. It's light. So if you illuminate chlorophyll by light, which is what we're going to see in a second, you're going to be able to make it fluoresce. So what's happening in these chlorophyll molecules? We have a light molecule that is going to be a photon that is going to hit the chlorophyll and these accessory pigments. The whole structure is called a photosystem. That photon is going to get bounce around to different chlorophylls. So they get bounced. Ultimately, that electron is going to hit a specific place. In this case, in biology, it's termed the reaction center. It is there at the reaction center that electron is going to get boosted into that higher energy state. And then they're going to get accepted by a protein. Okay, so now we need to get into the nitty-gritty of how this process actually works. So we're first going to go over it as this super duper cartoony idea, and then we're going to put it into a little bit more biological context. But I want you to sort of build on your understanding. So start at that overall light dependent reactions, light coming in, knowing what's coming in, what's coming out. Now we're going to add some concepts, then we're going to get even more specific with the mechanism. So we had that photon of light providing the energy to bounce an electron into a higher status. It's doing that in one particular location in the photosystems. So photosystems are the chlorophyll molecules and accessory pigments and proteins. So that electron with high energy is now going to get shuttled between different proteins, and it's going to lose energy as a result. It's going to go down an electron transport chain. And just like we saw in aerobic respiration, the cell is able to make something from that loss of energy. We get ATP out of it, and by we, I mean plants. So we're going to have a run of an electron transport chain. We're going to see in the future forming a hydrogen ion gradient and making ATP as a result. Now, just like in the electron transport chain in aerobic respiration, that electron needs to go somewhere. In the case of photosynthesis, there is another photosystem, photosystem 1, that is going to take that same electron and bounce it into a higher energy state. It's going to do that with the energy from another photon of light. That electron then still needs to get deposited somewhere, but it still has a lot of energy, and it's going to get deposited into that electron carrier. In this case, it's not NADH like in aerobic respiration, it's NADPH. There's an extra phosphate. So that was the super cartoon. We can put it into a little bit more biological context by again thinking about the photon of light bouncing around all these chlorophyll molecules until it reaches the reaction center. That electron then gets bounced into a higher energy state. But the electrons that had been in that chlorophyll molecule, they need to get returned. So they instantly need an electron. Where are they going to find an electron? What's always around? Water. So it's the exact opposite of what we saw in aerobic respiration, where you have an extra electron, you need to put it somewhere, put it on oxygen, make water. In photosynthesis, we need to steal some electrons. We need some electrons to be replacing the ones that got bounced up, and we're going to get those from water. As a result, you're stealing those electrons, you're going to produce hydrogen ions and oxygen. And I keep saying you, of course, you are not a plant. Okay, so what then happens to that electron? There is an electron transport chain that is going to pump hydrogen ions across that membrane, and we're going to be able to get ATP out of it, again from ATP synthetase. Continuing to follow the electrons, they're going to go to a new photosystem, get bounced again, up to a higher energy state with the energy from light, from a photon, bouncing around until it reaches that reaction center. And then that high energy electron is going to get deposited onto NADP plus to become NADPH. And again, there's an electron transport chain in this process. So I kept saying there's an electron transport chain. What does that mean? It's that shuttling of an electron, lowering the energy, and as a result, tricking hydrogen ions across that thylakoid membrane. Then, just like in aerobic respiration, there's a buildup of hydrogen ions in this inner membrane space. In this case, it's the thylakoid compartment. And there's only one door for those hydrogen ions to go out of. In this case, and in aerobic respiration, that protein is called ATP synthetase. It's like a giant rotating door, like those three-way doors that you circle in. And as those hydrogen ions pass and make that revolve, make it turn, that ATP gets formed from ADP and a phosphate group. So you can go through those steps. So again, you need to know the light-dependent reactions at this level to be able to follow those electrons, see where water gets made, how NADPH gets made, and how the end result, that ATP, gets produced. We can also think about that hydrogen ion concentration, of course, with the highest concentration of the gradient inside that thylakoid space and the lowest side of hydrogen ions outside in the stroma. So we have the products of the light reaction, ATP, that high energy NADPH, and of course oxygen. The first two, the whole point of those light reactions were to get that energy and those high energy electrons so that we can use them in the Calvin cycle to produce the glucose. Oxygen, well, that was just the byproduct. That was just the waste. We can even follow the molecules in water to think about what happened to the hydrogen ions. Well, those are the hydrogen ions that are building up in that thylakoid space that are ultimately going to flow back through ATP synthetase. Those electrons, well, that's what we were following. We were following them going through the electron transport chain after being bounced into that higher electron status. And the oxygen, that's waste given off. Which means now we're ready for the Calvin cycle. We're ready to take those products of the light reactions and see how they get utilized in the Calvin cycle. And in doing so, regenerate ADP and NAD+. So now we're ready for the light independent reactions, otherwise known as the Calvin-Benson cycle, or sometimes just the Calvin cycle. But we're going to see this does not involve light, so they're used to be called the light independent reactions. And it's here we're going to see that we're going to utilize that energy and the building blocks, carbon dioxide and water, to make sugar. So just like we saw earlier a few weeks ago, that coupling of endargonic and exergonic reactions, we have the exergonic reaction, taking that high energy ATP and NADPH, being able to produce that endargonic reaction, building up of that sugar. So that these are both high energy, and then we have the lower energy, the greater enthalpy molecules. Okay, so what do you need to know about the Calvin-Benson cycle? You need to know that there's an input of 3 carbon dioxide molecules. They are going to get fixed into organic molecules by an enzyme named Rubisco. Those molecules are then going to get rearranged, and I don't need you to know the names of all of the molecules, with the exception of G3P, but they're going to have this rearrangement. I'm not going to spend too much time on this metabolic pathway, other than to point out you are using the energy produced in the light-dependent reactions. In the production of the Calvin cycle, there's 3 phases where the carbon gets fixed in the first, so the carbon gets inputted into that organic molecule. It then gets reduced, but then we need to regenerate those products that were utilized in the very beginning of the cycle. So we need to regenerate RUBP, and so that's going to take more energy. I have this wonderful flash diagram I recommend you clicking on and sort of watching those reactions, but you don't need to know at the level of how those molecules are getting converted. So what you do need to know, you got the Calvin cycle. It's the light-dependent reactions, and it's what produces G3P. It produces this 3-carbon product. It's that G3P that's going to be making the glucose. So now prior to the last slide, I always said the product of photosynthesis was glucose, but like most things, that's an oversimplification. The product is actually G3P, which actually can be easily converted to be making lipids, to be making amino acids and proteins, or all those different types of glucose products, those polymers of glucose. So overall, what's coming in, what's coming out? But you actually need to know it at these 2 different levels, the electron transport chain, following those electrons, producing that high energy electron taxicab, and all that ATP, how from ATP synthetase and the hydrogen ions flowing down their concentration gradient. That NADPH and those electrons and the ATP are utilized within that Calvin-Benson cycle to take carbon dioxide and produce G3P. This is a great summary video. So I love this picture because what it's representing is the timeline of life as a road trip. So instead of thinking about time as time, you get to think about it as a distance, where 1 mile equals 1 million years. So if we start our road trip in British Columbia at 4.6 billion years ago, it takes a while for the Earth to cool down, but not too long after to actually have life have evolved by about 3.5 billion years ago. And prokaryotes were the only thing on Earth alive for the first 1.7-ish billion years. So prokaryotes rule Earth. Now, on this road trip, something amazing is happening right here in the equivalent of where we live, Phoenix. Approximately 2.7 billion years ago, we can start seeing oxygen increasing. Now, this production of oxygen by photosynthetic microorganisms, by photosynthetic bacteria, had an extreme impact on our Earth and the atmosphere. When oxygen actually started to accumulate, it completely changed the chemistry on Earth and allowed for the evolution of those eukaryotic organisms to be doing aerobic respiration like we saw last week, and allowed for the rest of evolution to have played out in the way it did. And so this actual oxygenation event fundamentally changed planet Earth like no other event. And it has allowed us that we can't actually go back now. That the chemistry that existed when Earth and life first evolved no longer existed after oxygen was released into the atmosphere in large quantities. And with that, we have finished the content for the Unit 2 exam. And I would highly recommend you check out the review materials.